Hi, and welcome back to our GCP-Mindset channel and all topics on clinical research. Today, we'll talk about Rule 9 of the responsibilities of the investigator according to good clinical practice. More after the break. Rule 9. Handle samples carefully and review all laboratory results immediately and accurately. Routine laboratory sampling, as a rule, is performed by study nurses at the site without any problems and followed by transit of the samples to your own local laboratory or to a central laboratory. Difficulties occur most frequently whenever unconventional investigations are carried out, for example, pharmacokinetic analyses, or if metabolites on dry ice need to be shipped to the central laboratory. Therefore, the investigator has to make sure that the responsible study nurse or other site personnel responsible is already present during the initial visit of the study site. The monitor needs to explain the handling, separating of samples, shipping, and labeling, including label marking. Correct handling and shipment of samples is essential for the safety of trial subjects and the quality of the trial data. Label samples carefully. Does it refer to the correct visit? And is it the correct subject identification code? Is it the right study code? In particular, when trial sites participate in several different studies performed by the same sponsor, confusion occurs frequently. If samples are labeled incorrectly, the database for subjects concerned will become incomplete, whereby, in the worst case, they will not be considered in the final data analysis. After the analysis of samples by the laboratory results are sent to the trial site. Usually this is done first by fax, followed by the transit of the original report by courier or post. The investigator should evaluate the fax report immediately upon receipt and should decide on which values are clinically relevant or how to continue treatment of the subject. Therefore, you should already sign and date the preliminary fax report so that it can be understood whether a control of laboratory findings was performed in a timely manner. Also, you should note in the laboratory report which measured values are clinically relevant to you and which are not. Clinically relevant laboratory abnormalities need to be recorded as adverse events in the CRF. Some values even fall into the category serious adverse event and must be reported to the sponsor within 24 hours after receipt of the test results. You should note, however, that not all laboratory abnormalities within the scope of a study are clinically relevant. Some measured values are not unusual for specific subject groups and therefore do not automatically represent an adverse event. However, an investigator must document that he reviewed each value and that he decided which of the abnormal results were considered to be clinically relevant. So much about the Rule 9 for investigators. We will look into each of the other rules in future videos, so stay tuned. We hope that we could give you some interesting information and look forward to see you next time. Goodbye.